membranes. A semi-permeable membrane is a membrane that allows certain molecules to pass through it by diffusion, facilitated diffusion, active transport, or passive transport. The rate of passage depends on the pressure, concentration, and temperature of the molecules on either side, as well as the permeability of the membrane. The membrane is a fluid allowing transporting things drift past each other. Phospholipid bilayer. The phospholipid bilayer is a thin polar membrane made of 2 layers of lipid bilayer. Here's your image of the phospholipid bilayer. Right here, these are the lipid tails, which are hydrophobic, which means they're water-hating. Right here, the blue things are phosphorus heads, which are hydrophilic, which are water-loving. And here are proteins, and they're there to transport substances. One of the ways nutrients and waste pass through the cell is active transport. Active transport is a movement across the cell membrane that requires energy, ATP. Substances are usually transported from an area of low concentration to an area of high concentration against their concentration gradient. Exocytosis and endocytosis are involved because they also require energy. Exocytosis is the expel compounds to the surroundings by fusing the membrane and then releasing the concentration contents into the cell's environment. Endocytosis is cells engulf large particles by extending their cytoplasm around the particle used to take in substances. Another example of passive transport is diffusion. Diffusion is a random movement of molecules from an area of high concentration to an area of low concentration. And here's your example.
Another way substances pass in and out of the cell is through passive transport, which is the random movement of particles from an area of high concentration to low concentration without ATP energy. An example is osmosis, the movement of water from an area of high concentration to low concentration. Here's an amazing drawing of water passing through a semi-permeable membrane, much like the cell membrane, to an area of high concentration to an area of low concentration.
Here's an image of the movements across the phospholipid bilayer. Active transport and passive transport pass through the proteins. Diffusion passes through the bilayer.
Throughout Chapter 3, we also discussed the different types of solutions. Hypotonic solutions. When a low concentration of solute and a higher concentration of water surrounds a cell so that the water diffuses into the cell, causing the cell to swell or even explode. Hypertonic solutions. When a high concentration of solute and a lower concentration of water surrounds a cell, causing the water to diffuse out of the cell, causing it to shrink or shrivel. And an isotonic solution is when there's an equal amount of solute and water inside and outside of the cell, causing water to diffuse in and out of the cell without a change in the cell's size. Here is a demonstration of a cell entering a hypotonic solution. There is much less water in the cell than there is in the solution, and as the cell enters the solution, it absorbs much of the water, it diffuses into it, thus resulting in the cell getting bigger. Here is a demonstration of a cell entering a hypertonic solution. There is many more water molecules inside the cell than there is outside of the cell. So when it enters the solution, the water diffuses out of the cell, resulting in the cell getting smaller. Another topic discussed throughout Chapter 3 is dialysis, the filtration of blood through a natural or man-made membrane. The first type of dialysis is peritoneal dialysis, which is the filtration of blood through the lining of your abdomen and a cleaning solution called dialysate. The second type of dialysis is hemodialysis, the filtration of blood through a man-made machine at the hospital.
Another topic discussed in Chapter 3 is how different types of solutions affect the cell. Plasmolysis. A higher solute concentration on the outside causes water to pass by osmosis from the cell into the surroundings. The effect on the cell is plasmolysis. The result of plasmolysis is a leaf that is wilted or limp. Cytolysis occurs when a cell bursts due to an osmotic imbalance that has caused excess water to move into the cell. Occurs in a hypotonic environment where water moves into the cell by osmosis and causes the volume to increase until the cell bursts. Deplasmolysis. The opposite of plasmolysis. Water rushes into the cell, causing the cytoplasm and plasma membrane to swell. The cell doesn't burst. The pressure exerted by the water on the cell wall is turgor pressure. Crenation. The contraction of a cell due to the loss of water through osmosis in a hypertonic solution. As a result, the cell shrinks and the cell membrane develops abnormal notchings.
Notchings!
Surface area to volume ratio. Cells are restricted to a size that ensures the surface area to volume ratio provides a sufficient membrane area to meet the needs of the cell. Surface area is calculated by multiplying the length times the width. In this case, all the sides are 2 centimeters long. So here you would multiply 2 times 2 and by 6 because there are 6 sides. So that gives you an answer of 24 centimeters squared. To calculate the volume, you multiply length times width times height. So here you would multiply 2 times 2 times 2, which equals 8 centimeters cubed. To calculate the ratio, you divide the surface area by the volume. So here you would divide 24 by 8, which gives you an answer of 3 to 1. Cells divide so nutrients can reach the middle of the cell, and so the cell can get rid of waste. This cell is considered an efficient cell because there's a large surface area and there's a low volume.